feet, or about 1,980 to 6,096 meters high. These are often made up of water droplets or ice crystals. The prefix alto is often used with the names of mid-level clouds. The low-level clouds form below 6,500 feet or 1,980 meters from the ground. These consist of mostly water droplets. Such clouds have the word stratus in their names. Finally, there are also vertical clouds. These are very tall clouds that can go across many cloud levels. There are 10 main cloud types. Cirrus clouds are thin and wispy and often seen during good weather. Cirrocumulus looks like tiny cotton balls, while cirrostratus clouds can mean rainy weather the next day. Altostratus have a dark grey appearance and are often seen on rainy days. Altocumulus clouds are small, white and puffy. Nimbostratus clouds are thick and grey and usually bring rain or snow. Stratus clouds are flat and grey and may bring about a drizzle, while stratocumulus clouds are low, puffy and grey clouds that may also produce light rain. Cumulus clouds are beautiful clouds, big, white and puffy. Cumulonimbus clouds are very tall clouds, spanning from low level to high level. These types cause violent thunderstorms with heavy rain and even tornadoes.
Hello everybody, welcome to all our grade 12 learners. It's Wednesday, so I'm Kathy Hasty and we are doing life sciences. Okay, but before we start, a huge shout out to Liberty. They are our major funders and they make all of this possible, and they are in it with you to the end of your matric finals to help you get through. Okay, people, don't forget to download your Tenfold education app. It has maths, it has science, it has life sciences. You can live stream, it's got a tutor function. So really use it, okay? It's there and it's also brought to you by Liberty and it's completely free. And then, you know how all these other apps have got in-app costs? This has got nothing. Liberty is trying to help you pass and do well in your matric finals. And then don't forget our WhatsApp number. We've got our little bot decks, and if you need exam papers, past exam papers, or you want to submit questions for us to go through here, for all the teachers here to go through, make sure you take a good photo, okay, of the or a good picture of the question, Otherwise, we can't use it. So we can only use really good questions and then submit it through Dex. And then, of course, we've got the YouTube channel. All you do is you go and you type in @MindsetLearn, and all the videos of us teaching here are there and available for you to use for revision. Okay, so without much further ado, here is the quote of today, or for today, our life sciences, as we do every Wednesday for life sciences. Don't stop when you're tired, stop when you're done. And that's Wesley Snipes, and what a gorgeous individual. But there you go, okay? So you don't stop when you're tired, you stop when you are done. And that's for your revision at the moment. And we're working towards your life sciences paper 2. So what I want to show you is the breakdown. For the paper 2 breakdown, your marks, they've changed, okay? Because remember, meiosis used to be in paper 1 and in paper 2. They've taken meiosis out of paper 2, paper 1, so it's only in paper 2. And it used to be 11 plus 12 marks, so 11 in paper 1 and 12 in paper 2. They've now just made it 21 marks. And then where evolution used to be 66, that's been reduced to 54, but they've also increased your genetics. All right, so please make sure you know your DNA, you know your meiosis, your genetics, and your evolution. And genetics and evolution, look at this. They make up more than half of your paper. All right, so let's look at our questions. There were lots of questions that were sent in, so thank you to all the learners who sent questions in. You are absolute stars, and I can see that you're working and you are revising. All right, so it was in the order that they came in, and I'm going to try and get through as many as I can. So, but at the same time explaining as I can. So this diagram, this was sent in by Majezu, and this diagram shows a cell during a phase of meiosis. So the first thing you are going to know is that it is going to be metaphase 1. And why is it metaphase 1? Because we have a double row of chromosomes at the equator. Okay? The labels you are always going to be asked is that there is the centriole. Okay? You have a centriole here and you have a centriole here. That set, the double chromosomes here, are homologous chromosomes. Now remember, homologous chromosomes are the same length. They're the same length and size. They have a centromere, which joins the two chromosomes. So let me just put this as a label as well, because they also ask that. It's a centromere. And please don't confuse centriole and centromere. A centromere, think about that it merely holds the chromatids together. So that's why it's called a centromere, okay? Holds the chromatids of a chromosome together. Homologous chromosomes are the same size and length. They have a centromere in the same position, and they carry the same genes in the same positions or loci on the chromosomes, okay? This here, one of them would be a paternal chromosome, and one would be a maternal chromosome. And you can see crossing over has occurred because there's a piece there and that of this one, and there's a piece of this one here. Okay, so those are homologous chromosomes. Oh, and then they also ask you spindle fibers. So they're all the labels that you need to know for a diagram on meiosis. Okay, remember prophase, you're going to see crossing over in prophase 1 and no crossing over in prophase 2. In metaphase 1, you'll have a double set of chromosomes. okay, lining up. So you've got your maternal and paternal chromosomes lining up at the equator, whereas metaphase 2, there's only a single row. You've got individual chromosomes lining up at the equator. Anaphase is for away. So the chromosomes are being pulled away from the equator towards the different poles, all right? And anaphase 1, they look like little butterflies because you've got whole chromosomes. And in anaphase 2, you've got chromatids being pulled across. And then telophase 1, 2 cells. And telophase 2, you've got 4 cells. You must be able to recognize the phases. Okay, so give labels for A and B. We've done that. Identify the phase. We've done that too. It's metaphase 1. Okay. Oh, give a reason for your answer. So why is it metaphase 1? Well, easy, because we've got homologous chromosomes. And you don't have to call them homologous chromosome pairs. Homologous chromosomes are a pair. One is you've got a paternal chromosome and a maternal chromosome. If they are homologous chromosomes, they are a pair of chromosomes. So homologous chromosomes are lined up at the equator. Or you could say there is a double row of chromosomes at the equator. Okay, then explain why this is a diploid cell. Now, if we look here back at this diagram, why is it a diploid cell? Because it has homologous chromosomes. If it had already undergone reduction division, you would then have individual chromosomes, which would be at the equator, and they would then end up with chromatids at each cell. So, which would then, or chromatids, which would become daughter chromosomes. So, why is this a diploid cell? Well, that's easy. There are 2 sets of chromosomes present. One maternal set and one paternal set. So one from the mom and one from the dad. Okay? Because karyokinesis, which is the splitting of the nucleus, karyokinesis has not taken place. Okay, so remember, karyokinesis is when in meiosis you've got you've first got reduction division and then you've got duplication. So with meiosis 1, you start off with a diploid cell, and then in meiosis 1 it becomes N. So that's reduction division, and then this will then duplicate so that you end up with 4 unidentical daughter cells. Okay, so let's go back here. How many chromosomes will there be in each gamete produced by this cell? Well, if there are 2 sets of chromosomes, then there are going to be 1 set of chromosomes. So what do we have here? Here we've got 4 or 2 pairs of chromosomes, which means we'll end up with only 2 chromosomes. Because we had 2 pairs, which is 4, and that 4 is then going to go into 2 and 2, and 2 and 2 and 2 and 2 for each of those gametes produced. Explain the importance of genetic variation in the process of evolution. Okay, let's just quickly remove this. Okay, so if we're going to talk about genetic variation, remember, my pen's doing funny things, variation results from prophase 1 and metaphase 1 and 2. Okay? This ensures that offspring with favorable conditions, or favorable, because remember they're talking about evolution, which is natural selection and survival of the fittest, and genetic variation. So firstly, where does genetic variation come from? From prophase 1 and metaphase 1 and 2. Okay? And what does it ensure? It ensures that the offspring with favorable traits, you can always write traits instead of characteristics, it's the same thing. Traits survive to reproduce, okay, while those with unfavorable genetic traits, which result from genetic variation, genetic traits die. Okay, that's the basics of Darwin's evolution and natural selection. So those with the favorable traits, they survive and reproduce, while those with unfavorable, okay, genetic traits die. Okay, this results in natural selection and survival of the fittest. I mean, how easy is that? So you've got to remember genetic variation. Every single organism on this planet has some form of genetic variation, okay? And that genetic variation, some traits are favorable and others aren't. So those organisms with the favorable traits, they survive and they pass it on to the offspring. Those that don't have the favorable conditions, well, they die. And well, then there's nothing to pass on to anyone, okay? That is what natural selection is. It's survival of the fittest. All righty, let's see our next question. Okay, here we've got Mjezu as well. The diagram below shows the karyotype, which means all the chromosomes of an individual with a disorder, okay, caused by non-disjunction. Okay, now non-disjunction is when chromosomes do not separate during metaphase 1 and 2. So what does this diagram here tell us? Okay, let's go. First of all, what do we have? These are, that would be, let's say, a paternal chromosome. This would be a maternal chromosome. So this is a diploid organism because I have two sets of chromosomes. They are homologous chromosomes. Okay, and as we've said, they are the same length, they're the same size, they have their centromeres in the same position, and they carry the same genes in the same locus. Okay, that makes them homologous chromosomes. Chromosomes 1 to 22 are called autosomes. And they are responsible for all your characteristics, except the sexual ones. And then chromosome pair 23, these are your sex chromosomes, or you can call them gonosomes. Okay? And in a male, this will be a male, it's XY. And they are non-homologous chromosomes. Okay, now, the fact that they are non-homologous chromosomes, can you see that they're not the same size? So this chromosome here and this one don't carry the same alleles. This one's going to have a whole lot less alleles. If I stand corrected, but I think the Y chromosome only has about 38 or 39 alleles, whereas X has a whole lot. And in the female, we have XX gonosomes or sex chromosomes, and they both look like this one, and they are going to be homologous. And that's important for when you do sexual disorders that are carried on the sex chromosomes, which is the X chromosomes. Okay, then name this disorder. Okay, let's just have a look. All right, our disorder here, do you see that this chromosome 21, there are 3. There's 1, 2, 3, when there should only be 2. They should be homologous chromosomes, which means pairs. So if there are 3, this is called trisomy 21, which means that there are 3 copies of chromosome number 21. So the disorder that's caused here is Down's syndrome. Okay, then the phase in meiosis when non-disjunction occurs. Now there are actually 3. It can occur during, 2. It can occur during anaphase 1 or anaphase 2. Okay, so let's just quickly zip down here. If I have anaphase 1, you're going to have okay, so your chromosomes are now going to be pulled. You've got your, this is anaphase 1. Your chromosomes are going to sit here like this. They look like little butterflies or little bows. I always think of them as little butterflies. This is normal. Okay? But when non-disjunction occurs, now disjunction means to separate. Non-disjunction means that they don't separate. So we end up with 2 chromosomes being pulled onto one side, and I almost used my finger to rub this out. I'm really losing it. Okay? And there's going to be nothing here. Okay, so what happens? Now I sit with, at the end of meiosis 1, so telophase 1, I now sit with 4 chromosomes and 2 chromosomes. If it occurs in anaphase 2, okay, so remember this is happening in 2 different cells, the same thing. So I'm going to have my single set of chromosomes here at the equator, all right, and now they get pulled up. So what happens? This one pulls that way, that one pulls that way, this one this way, and that one that way, so they look like this. Because you've got a single row of chromosomes. So I now sit with a little boomerang this way, and this would be normal. But what happens when there is non-disjunction? What happens? Well, it doesn't have to be on this side. I'm just drawing it on this side. We end up with 2 being pulled this way. So a whole chromosome gets pulled this way, and this one ends up with nothing. So the result here is going to be a cell that has 1, 2, 3 chromosomes and one that only has 1. So anaphase 1 or anaphase 2. Alrighty, then the type of mutation that results from non-disjunction, what are we going to call that? It is now they want to know the type of mutation. There are only 2 types of mutations that you need to know. There's chromosomal mutation. Why? Because the chromosomes, there's a problem with the chromosomes. You've got 3 chromosomes sitting at, or 3 chromosome 21s, okay? And that's going to be trisomy. And the other cell that results is going to be monosomy, which means it has 1 less chromosome than it's supposed to have. And those embryos never ever make it. They don't even get to be embryos. The body just aborts it. Okay, so chromosomal mutation. Remember, the other mutation you get is a gene mutation. And a gene mutation is when the DNA triplet, when one of those nucleotides is different. So that changes the gene. Okay, and when it changes the gene, well, then you have other issues that happen, especially during protein synthesis. So when would a gene mutation occur? It would occur during DNA replication, or it would occur during transcription when DNA makes RNA. Then you can have a problem there. But it's a problem with 1 nucleotide in that gene sequence, okay? Whereas a chromosomal mutation, you sit with it's either got 1 too many chromosomes or 1 too few chromosomes. Okay, now identify the type of chromosome numbered 1 to 2. Oh man, they're autosomes. Okay, they're autosomes, and remember 23 is either XX, it's female, they are homologous, and they are your sex chromosomes or gonosomes, and in males, XY, sex chromosome or gonosomes. Okay, I think before we do our next question, let's go for a quick ad break so you can go and get some tea or do whatever it is that you do, but very quickly, and I'll see you all back here in a minute and a half.
Did you know that a full head of human hair is strong enough to support 12 tons? We have on average about 150,000 hairs on our head. According to research, the molecular structure of hair is such that a single strand can support about 100 grams. This means that the combined hair on the average human head would support over 12 tons, which is about the weight of 2 African elephants.
No one ever asks why we do the weird things we do. Like, why once is never enough. Or doing 2 trips is just 1 too many. Why do we, before we use these, or say 5 when we mean 20? Why do we have second thoughts? And think that lift buttons make them go faster? Why don't we walk there? Cross our fingers, touch wood, and struggle to hide our true feelings? At Liberty, we want to know all the weird and wonderful things that make us human. Because human beings, being human, are what we make financial solutions for. Liberty, in it with you.
Hi guys and girls, we are about to carry on, but before we do, a huge shout out to Liberty, in it with you so that you can be the best you can be. Then don't forget to download your Tenfold education app. It's got lots of goodies for you, so download it. It's also brought to you by Liberty. And then our WhatsApp little Dexy boy, you can send him a WhatsApp if you need question papers or if you want to submit question papers, all right? And then, or questions for us to do here. And then don't forget YouTube. You go at Mindset Learn, and videos of what we do here is posted up on YouTube. You can access it there. So, and also everything brought to you by Liberty. So yeah, Liberty, you guys rock. All right, now our next question is brought to you by Karabo Princes. Okay. And the diagram below represents the evolution of flightless birds and the continents on which they each represent. Now, okay, I've got some space here, so I just want to tell you this. The minute they talk about continents, they are referring to continental drift. So, the theory is that a long time ago, all the continents were just one big continent called Pangaea, which means one Earth, one continent, okay? I can't remember the exact translation. So then you had Pangaea, and then Pangaea split. And Pangaea then split into Gondwana land, okay, which is in the south, and Lauren, let me just, I'm trying to get the spelling correct. Laura, oh, for heaven's sake. And Laurasia, there we go, Laurasia. Okay, and this clearly is the whole North America and Asia, and this was in the north. All right, now, if you look at these, Africa, South America, New Zealand, Australia, and well, New Zealand isn't a continent, but okay, we'll forgive them for for saying it is, but it's actually a set of islands. But you've got Africa, South America, Australia, and South America again. Those all were part of Gondwana land. And then Gondwana land split into Africa and South America and Australia, okay? So if we look here, we have a common ancestor of flightless birds. This, by the way, is a phylogenetic tree. And what a phylogenetic tree does, it's like a family tree, but over a very long period of time. So your time will run across here, but they haven't put time in. So time will go along here. They haven't put the time in. So look, we have this common ancestor that then we ended up with the ostrich and this common ancestor here. So what do we end up with? We end up with a rhea, which is going to be quite close to this one. So I'm going to make that a 1.5. And this one here, which then becomes the common ancestor of the kiwi. So we'll make that 2.5. And it again splits here. So we'll make that 3 into 3.5 and 3.5. So who are the closest related here? It's going to be the emu and the cassowary. All right, so the cassowary in South America and the emu in Australia. Right, why? Because they had the most current common ancestor here. Okay, the ones that are the furthest related would be the ostrich and the emu and cassowary. Why? Because look how far back the common ancestor was here. Okay, so what do we see here? Identify the types of diagrams shown above. I told you, it's a phylogenetic tree. Or you can also call it a cladogram. Okay. But philo means the different phylums, and genetics. So they're all different organisms that belong to the same genes, or the same, that have relatively the same genetic tree. Okay, name the two species that share the most recent common ancestor. Well, we said it was the emu and the cassowary. You see, they are here. So the emu and I can't think of how to spell this. And don't spell it wrong when you got this in the exam. Cassowary. Okay, make sure that your spelling is accurate. All right, now let's look at our next question. Use the information in the diagram to describe how biogeography supports the theory of evolution. Okay, so now remember, what do we have? We've got a flightless common ancestor existed on one continent. Okay, now you can actually turn around and you can say that continent was Gondwana land, okay, or you can just say on one continent. Continent, all right. I'm going to put in brackets here called Gondwana land, okay. Then continental drift, caused the original ancestor or original population, which was the common ancestral population, the original population to be split, okay? Separated by the ocean. Okay, now each population, I'm just going to write pop, each pop evolved independently. Okay, so independently, the ostrich in Africa, the rhea in South America, the cassowary in South America, the kiwi in New Zealand, and the emu in Australia. Now, you must remember that the climates and the environmental conditions in Africa and South America and New Zealand and Australia are all different. So, what happens? We have, I'm going to run out of space. Each population evolved independently. Why? Due to different environmental conditions. Okay? And remember the variation we spoke about just now? And natural selection. Okay, do I have enough space here? Yeah, okay. So they became genotypically and phenotypically different. Okay? Over time. So each continent now has different species of flightless birds. And you know, people, all you've done is you've explained what you see here, okay? We had all of these sitting on Gondondwana land. You had the common ancestor, which was now split because of continental drift. So Gondondwana land split into those different those different continents. What do you have? You have different environmental conditions, and therefore natural selection occurred, and you end up with those with favorable conditions surviving and those with unfavorable conditions dying. So what is that? That's natural selection. So that after a long period of time, they are genotypically and phenotypically different. And can they interbreed? No. Can they produce, maybe they can interbreed, but can they produce fertile offspring? No, which means they're different species. Okay, easy peasy peasy. You must make sure you read your questions and describe how it can be proven that ostriches and rheas are different species. Give them time to interbreed. And if they do and produce, and this is the clinker, and produce fertile offspring, then they belong to the same species. Now remember, what is a species? A species is a group of organisms that look similar, okay, that have the same DNA sequencing, all right, that can interbreed and produce fertile offspring. All right, what's a population? A population is a group of organisms of all different ages, all right, that live in the same area, that they live in the same area, they interbreed, and they produce fertile offspring. So if they live in different if they live in different areas, they are going to evolve differently because of natural selection. You know what else? You could also say, or you could do DNA sampling and analysis and compare the ostriches and rheas. Okay, so that DNA sampling, you're going to do a PCR test just like they're doing with COVID. Okay, how are we going for time? Okay, guys and girls, let's go. Okay, now we've got Ntombi Kayise, I'm just going to say Ntombi's question here. It's about cloning. Okay, so the first cloned animal in Africa. By the way, we are all very proud of Futi. Futi is a little cow, and she was actually a main feed. She was born in Northwest Province in South Africa on the 19th of April 2003. Okay, no fertilization took place. Why? Because it's cloning. Okay, she was produced from a single cell from the ear of a donor cow named LMJC865. That's actually irrelevant. The donor cow, this is what is relevant, has a high or had a high average milk yield. In other words, this was a trait to meet human needs. That's why we have genetic engineering. Okay, remember, genetic engineering involves GMOs, it involves cloning, it involves creating GMO products, cloning, and also stem cell research. All of that is genetic engineering. Why? And using artificial selection. Why? To meet human needs. We humans want what we want, and we don't care what we do to get there. Okay, so cloning allows for the production of organisms. Now, this is an advantage for production of organisms with desired characteristics. So that would be an advantage. And some people argue that cloning reduces genetic variation. We don't have to argue, it does. Okay, so this would be a disadvantage, and you must know at least 2 advantages and 2 disadvantages of cloning. In the offspring, there's no further genetic improvement. Why? Because your gene pool is limited. Cloning is very expensive, okay, and may not be economical for commercial agriculture. So you could do it for the odd cow here and the odd cow there, or the odd sheep, or the odd whatever it is you're going to clone, but you can't just do it for an entire herd. It's just too expensive. Okay, so according to the extract, an advantage of cloning and a disadvantage. Okay, people, I'm not writing this out. An advantage would be it allows for the production of organisms with desired characteristics, or you could say traits, and a disadvantage reduces genetic variation, or you could put expensive procedure, or you could put not commercially viable for agriculture. So there you go. Then state why the donor cell was taken from this cow and not from another cow. Well, guess what? This cow was selected because she carries the genes for a high average milk production. And let's not wait there. Let's go. I mean, it's only 1 mark, but you could say milk production of 78 liters per day. But really, this is what she was selected, because she carries the genes for a higher than average milk production. That is why she was chosen. Okay, they want to create another cow that can produce milk like this. Okay, so state why an ear cell was used and not an ovum. All right. Now, what do we have? First thing, now you've got to be able to explain an ear cell, why that was chosen, and why an ovum was not chosen. So, easy peasy. The ear cell is diploid and carries 2 X chromosomes to ensure a cow, or a female offspring is produced. Okay, remember, an ovum in a female only carries XX, but in a sperm cell carries X and Y. Okay, so this ensures a female offspring is produced, all right, and with the desired female offspring is produced with the desired traits. Okay, an ovum, remember, 1 ovum, many ova. The ovum is haploid and only carries half the genetic information. Whereas this diploid ear cell carries all of it. Okay, it's diploid, X chromosome. I'm going to put your and the genetic info. All right, whereas to ensure that this female is going to carry everything, those desired characteristics. Now, briefly describe the process of cloning. I'm going to talk you through this because it's far too much to write. Okay, so here we go, cloning. They have a surrogate cow. Okay, that surrogate cow is now ovulating. So they remove the ovum the haploid ovum from the surrogate cow. Then they take any somatic cell, an ear cell, an udder cell, it could be a cell anywhere in the body from the donor organism. In this case, it was Futi's mom. All right, so they take it from the donor cow and they take that diploid cell nucleus. The ovum, they remove the nucleus and chuck it away. And they insert that diploid nucleus into the ovum. Now the ovum is a diploid zygote. Okay, although fertilization didn't occur, it's got a diploid nucleus in it. They then use electric impulses to stimulate mitosis so that this ovum, this diploid zygote, can now undergo mitosis and grow and develop into an embryo. When it gets to an embryo or a blastocyst stage, they then take that blastocyst, the embryo, and they implant it into the surrogate, the surrogate female's uterus, where the lining, the endometrium, is all ready to receive a new little embryo, okay? And it then grows and it develops, and that surrogate female gives birth to a new offspring. The offspring is genetically identical to the original donor organism and not the surrogate organism at all, okay? You must know the process of cloning. Okay, we're on to our next question. Okay, oh, this is also from Ntombi. All right, there are, oh, wait. They're telling me there was another part of a question here. Oh, here we go. All right, so here we got genetics, yay. Okay, the flower color, purple or white. Alrighty, now people, there's your first clue. If it's purple or white, they are telling you that this is complete dominance. If they said purple, mauve and white, then it would have been incomplete dominance. And if they said purple with white markings, you can end up with codominance. Okay, so the fact that they say purple or white, it's complete dominance. Okay, in a particular plant species controlled by two alleles, D and D. Okay, 4 crosses were carried out to determine which allele is dominant. 40 offspring were produced in each cross. The phenotypes, which is what you see physically of the parents, and the offspring in each cross were recorded. So here are our crosses, parent, parent, offspring. Now, when we had purple parents combined with a white parent, we ended up with 40 purple offspring, which means these offspring are heterozygous, and therefore they are going to be capital D, small d. Which means, and the fact that they are all purple, based on Mendels Law of dominance, this purple has to be DD, and the white is going to be recessive, lowercase d, lowercase d. When I have purple and purple, I have 31 purple and white. Therefore, what have I got? I've got a 3-to-1 ratio, which tells me this one is heterozygous and this one is heterozygous. And when I cross them, I'm going to have 75% will be purple, and we're going to end up with 25% are going to be white. You must know your basic crosses. Then a white and white gives me all white. That's because white is recessive, and purple and white, the fact that we end up with white offspring means this purple is heterozygous, and we know that white, in order for the recessive trait to come through, you always need 2 recessive alleles, always. 2 recessive alleles for that recessive trait to come through. But if it is a dominant allele, then when it is homozygous, well, clearly it will dominate. It is that color or that trait. And if in the heterozygous state, the dominant characteristic or trait will come through. All right, so now that we've analyzed that, let's look at our question. State the dominant color, easy, purple. Okay, be careful, they may trick you in an exam. So let's say, for example, they said red and white, but they made white dominant. Be careful, look at the question every single time. Don't assume because red is darker or black hair is dark, dark and white hair is not, that white is recessive and black is dominant. Don't always assume that. Okay, use cross 1 to explain your answer here. Okay, easy. When we used, no man, when we, and I'm going to just talk it through because we are really running out of time. So what you say here is when a purple parent or a purple flowering plant, DD, was crossed with a white plant, what do we end up with? Okay, we end up with offspring that are all purple. So those offspring are all heterozygous, which applies to Mendels Law of Dominance. Okay, then, state Mendels Law of Segregation. Easy, easy peasy. When two alleles, and I'll show you now how easy this is. When two alleles for a trait or characteristic separate during meiosis, okay, because remember we need meiosis, we've got to get gametes. So that when two alleles for a trait separate during meiosis, what am I doing? Each gamete will only contain one allele for the trait. So let me show you. Let's do it quick sticks here. Okay, let's take a heterozygous so we can see. So we do DD. Okay, now meiosis takes place. So meiosis 1, what do we have? We have D and D. And then during meiosis 2, we have D, D, and capital D and capital D. Okay, so one of these little gametes, these are all your gametes. Okay, that's how it works. And that's all they're saying is two alleles, which is the capital D and the lowercase d, for a trait separate during meiosis so that each gamete contains only one allele and not both. If it contained both, then obviously it's diploid. All right, use a genetic cross. Okay, I'm going to do this very, very quickly so that we can finish this question. Here are where your marks are going to be. They say the two purple flowering plants shown in 2, let's just quickly check, 2 was purple and purple heterozygous. Okay, so now we have a heterozygous cross. So we're going to go P1, and the phenotype, I'm not going to write it, so phenotype will be purple times purple. Okay, the genotype is going to be capital D lowercase d times capital D lowercase d. Then we have meiosis occurs. So that's 1 mark, that's 1 mark. This is a half a mark. This is a half a mark. Okay, so what do we have? Meiosis occurs. Here's the Punnett square. It's got 9 little brackets. So capital D, lowercase d, capital D, lowercase d. We have our gametes. Okay, and this fertilization takes place. Fertilization. Okay, that's another half a mark. Okay, so your gametes, that will be capital D, that's homozygous purple. Capital B, lowercase d, heterozygous purple. Capital D, lowercase d, heterozygous purple. Lowercase d. What is my ratio here? So my F1, what do I have? I have the genotype. Please, you must write it out. Hey, genotype, I'm going to have 1 to 4 is going to be capital D, capital D. We're going to have 2 to 4 is going to be capital D, lowercase d, and 1 to 4 is going to be lowercase d, lowercase d. When we do the phenotype, okay, by the way, this is another half a mark. So that's another mark there, and your phenotype is going to be 75% are going to be purple, and 25% are going to be white. They're your little whiteys. Okay, end of story. Easiest, oh, that's another mark. That's easiest 7 marks you've ever got. Okay, so please make sure that you know that format. Go back, check where the marks are. All right, so people, remember you're writing on DNA, meiosis, genetics, and evolution. Okay, there are only specific things that they can ask you. So make sure that you know those specific things. With DNA, you must know the structure of the DNA, the structure of the RNA. You must know transcription, you must know translation, and you must know DNA replication. For meiosis, know the different phases. Okay, please be able to identify them and know when crossing over takes place and what allows for genetic variation. As far as genetics is concerned, make sure you know your crosses, make sure you understand the sex determination and sex-linked diseases and chromosomes, okay? And dihybrid crosses and monohybrid crosses, and pedigree diagrams. And then for evolution, and I've run out of time, so just learn, okay? Know your work. We've gone over this stuff 1,000 times. Have a wonderful week. Stay safe. Take care. Mwah.
What's up, peeps? Welcome to Moby School. We are here to make sure that you, yes, you get all the marks that you deserve on the go. We're here every Mondays to Fridays from 2 to 3 p.m. only on Mindset Learn TV. Now, this is a whole hour worth of revision just for you. And